Today we'll be focusing on, it's sort of a two-part webinar. So I to first focus on our GlycanFinder software and give an overview of the key features of GlycanFinder. And then next we'll dive into what we've been working on in our BSI lab services, in more particular, the analysis of purified glycoproteins, and also looking at characterizing glycopeptide enrichment data. And so the key features of GlycanFinder, as I mentioned earlier, is to identify both N and O-link glycans and do glycan profiling with structural resolution. And we could do glycan profiling at specific sites within proteins and its support for multiple enzyme digestions. We could do quantitative analysis, and we support both label-free and labeling methods such as TMT. Glycan de novo sequencing is employed for finding unknown glycans that are not present in your glycan database. And it comes with a user-friendly graphic user interface. And we support Orbitrap data, timsTOF data, as well as Xenotoph data. So as you can see here are various spectrum from different instrument types as well as different fragmentation types. So here we have HCD fragmentation from Orbitrap, which provides very good peptide backbone fragmentation, as well as glycan fragmentation. We also support timsTOF CID data, again, very nice glycan fragmentation, slightly lower level peptide backbone fragment ion intensities. And then for Orbitrap ETHCD as well as Xenotoph EAD, you really get very rich spectrum, not just B and Y fragment ions, but also C and Z fragment ions. And these two types of instrumentations and fragmentation modes really help to identify glycosylation sites and also produce good glycan fragment ions for glycan identification. And so within GlycanFinder, the algorithm workflow is as follows. So starting with the raw LC-MS data, we first perform a peptide-based search. So we try to match the fragment ions in the lower M/Z range of the spectrum to protein sequence database. And then we also search a glycan database, trying to match the glycan fragment ions. And then if we have a good match for both, we've identified a glycopeptide. However, if that's not the case, we can still perform a glycan-based search. And so we still search the glycan fragment ions against the glycan database. And even though maybe we don't have good peptide backbone fragment ions, we can still retrieve a potential glycopeptide. Then all of these results are controlled by an FDR. And then lastly, if we still don't have any matches to our glycan-based search or peptide-based search, we can perform glycan de novo sequencing to retrieve anything that's not found in your database. If you'd like to know more about the algorithm and how it works, please refer to our paper, which was published in Nature Communications last year. And so what's new in the field of glycoproteomics and in analyzing glycoproteomic data is the fact that we're able to identify both an N-link glycan and an O-link glycan within the same peptide. And so here we're showing peptide where we have an N-link glycan at the very end terminus, as well as an O-link glycan at position 3. And so if you can't differentiate or have the ability to identify both N and O within this peptide, you may result in a false assignment of just one glycan that has the same mass as the mass of these two glycans combined. And the differentiating factor, as highlighted here, is this Y11 ion that's still associated with a glycan moiety, which we're able to use to differentiate these two glycosylation sites. And so site ambiguity is a big challenge within the field, and especially with O-link glycans. So as I mentioned, they can occur at really any serine or threonine, And so there are a number of cases that can occur within peptides that are rich in serine and threonine residues. So within the first case, and the most ideal case, is observing fragmentation ions that flank the individual residue that's glycosylated. And in doing this, we have the ability to exclude all but one site within that peptide. But of course, this isn't always the case. And the other, I guess, most simplest form is the ability to exclude all other sites based on there only being one potential glycosylation site within that peptide. So within this peptide, there's only one serine, so it would be assigned to that serine. Oftentimes, we don't get complete fragmentation of the peptide backbone, and so we can not always exclude every other site. And so we're left with some ambiguity. And then lastly, if we can't localize it at all, we just have an inability to assign it to a specific site, and the glycan site is ambiguous within that peptide. And so to provide users some confidence in assigning glycosylation sites, we provide what's called A score. So A score represents the probability that that glycan occurs at that site and not any other site within that peptide. So here's an example of by analyzing the glycan fragment ions, and A score is important. So here we have the same scan and the same peptide sequence and a very similar glycan mass. So the question becomes, which one is a better match to this spectrum? And within GlycanFinder, we provide these spectral mirror plots. So on the top, we'll show the fragment ions that match to this best scoring glycan on the top of the spectrum. And below is the second best scoring match that these glycan that we can match to the glycan fragment ions in the bottom part of the spectrum. But what you can see when we zoom in here is there's a number of unique glycan fragment ions that can only match to the top scoring glycan and not the second best match. And so we use this information to help guide what the glycan identification should be. And we can also use it to define what the glycan structure should be. And so we also provide structural resolution within GlycanFinder. So here are two glycans that are isomers of one another. They have the same glycan composition, just slightly different structures. So again, here's the mirror plot of the same spectrum. And on the above is the top scoring isomer that matches, and highlighted here are the glycan fragment ions that can only be matched to the isomer shown at the top of the spectrum. And so like A score, we provide what's called an S score for our structural resolution. And this provides the user confidence in whether or not we can differentiate between these two structures given the spectrum and the data at hand. And so GlycanFinder has two main workflows, the first being glycan site profiling. In this workflow, we accept multi-enzyme data, we perform the database search, and could do obviously site profiling. The second main workflow is more for quantification. So again, it's mainly done with just one particular enzyme so that the data is comparable across samples. And then we perform a database search, and you have the option to do label-free quantification or select from a labeled method such as TMT. So here's an example of glycan site profiling data. Here we have a human IgG, and it's highly glycosylated. And so you can see we have highlighted all the asparagine residues as well as threonine and serine residues that are glycosylated. And by simply clicking on these residues in the protein coverage view, we can pull up this pie chart to look at relative abundances of glycans at the specific sample level. And we also have another site profiling tool within the same workflow. So for any protein, glycoprotein you've identified, we can use the site profiling to look at each individual glycosylation site, which can be selected from a drop-down menu. And also we can select specific glycans that we want to compare and analyze, and which samples we want to compare. And so here you can see at position N180 in this IgG1 protein, we can compare the abundance of glycans across two samples. And then, as I mentioned, we support label-free and labeled methods. So because we do feature detection of glycopeptides, we can extract the area values from the area under the curve of the precursor ions in order to do label-free quantification across samples. And also, we support TMT analysis. And so here you can see we can compare relative ion intensities from the reporter ions of TMT tags. Okay, so as I mentioned earlier, we can also retrieve unknown glycans that are not found in a database search. And so the way we do this, and it's described in more detail within our paper, but we first build an N-link glycan tree, and this is constructed from the N-link core structure by iteratively adding monosaccharides to that tree. And at each of these iterations, we employ a dynamic programming algorithm, which is coupled with a graph transformer neural network and used to predict the next monosaccharide based on the input spectrum and the partial tree from the previous iteration. So we keep going along this spectrum, adding different iterations until we've come up with the final glycan identification. And so here's sort of a case study where we searched the same data first with a restricted N-link database of only 15 glycans, and then searched the same data set against a more comprehensive N-link database. And so, of course, we identify far fewer glycopeptides with the restricted glycan database than we do with the more comprehensive one. But what I wanted to show is that we can retrieve these glycans that were not in the database to begin with, which are found in the more comprehensive database simply by using our glycan de novo sequencing function. And also, if you identify these glycans that we have high confidence in, they have good glycan scores, we can add these into the glycan database for future searching. So we have a number of built-in glycan databases within GlycanFinder, and you can go in and select any of these glycans and choose whether to modify the existing glycan or add it in as a new glycan. So it's very user-friendly. You click on the monosaccharide you want to add the next one to, or you can remove some and build your specific glycan and then add it to the existing database or modify the existing one. Okay, and lastly, I wanted to mention BSI and our team has joined the Human Glycoproteomics Initiative for the second part of the study. So we weren't part of the first study, which is published in Nature Methods, shown here, where they evaluated a number of different software platforms from the software development aspect as well as the user aspect. And they employed a number of different criteria that score the accuracy and sensitivity of both N and O glycan identification. And so the same sample was run across all platforms and from all users. And then the authors of the study and HGI study scored the results based on these criteria. So we participated in the post-study benchmarking. And so shown at the bottom here, so the score goes from 0 to 1, 1 being the highest score you can achieve. And so for N-link glycan analysis and O-link, here are scores from GlycanFinder, and they actually are slightly higher than the next best team, which was MS Frager, from the post-study benchmarking analysis. Okay, so I'll go into what we've been working on within the BSI lab now. And first, going over some analysis of purified glycoproteins. And so we chose a couple different glycoproteins to do method development and optimization with, the first being tirnosep, or otherwise known as Enbrel, which is a highly glycosylated and soluble dimeric fusion protein that consists of the ligand binding domain of tumor necrosis factor receptor fused with the Fc portion of IgG1. And this functions as a TNF inhibitor by competitive binding and prevents activation of the inflammatory response, and is licensed for treatment of autoimmune disorders such as arthritis. And so we, along with our partnering company Bison Bio in China, we developed workflow to analyze purified proteins and extract, the most number of glycans we can, but also attempting to localize the most number of glycans within this protein. And so we have both an N-linked and O-linked workflow. They both involve screening for the predicted glycosylation sites within the protein and selecting specific enzymes that would give the best digestion for the analysis. With the N-linked workflow, we do reduction alkylation, multi-enzyme digestion, desalting, and run on our Orbitrap in both HCD and ETHCD mode. For O-linked, again, we do the O-linked glycosite prediction. We remove the N-linked glycans with P and GAs-F, and then we perform sequential digestion with IMPA, which is now known as O-glycoprotease, which cleaves at right before mucin type O-linked glycans, and then did a trypsin digestion following that. And with the O-linked workflow, we either then do desalting and run the instrument, or do desalting followed by glycopeptide enrichment and combine all of this data. So we've done a lot of method optimization with our instruments, and we've kind of settled on two different instrumentation methods, one being ETHCD. As I mentioned earlier, this provides those C and Z ions, helping to localize glycosites. But what's important is we use a targeted mass trigger. So anytime we see a mass corresponding to one of these oxonium ions, we can send the precursor for ETD fragmentation, shown here. And then we also use just a straight HCD method, but we use step collision energy. So for each precursor, we use step collision energy of 20, 30, and 40%. And so from all of this data, we were able to identify over 1,700 glycopeptides, 451 N-linked glycans, 1,251 O-link, and 23 glycans with N and O-link. And overall, we achieved over 97% coverage of the Enbrel protein. 25 sites passed our A score of 20, which is equivalent to a p-value of 0.01. And there were only 14 previously reported, so we actually identified 11 new glycosites within this protein. And we see a nice distribution of N-linked glycans across all three of the N-linked sites within Enbrel. And also the site profiling data revealed that we have good consistency in our 3 replicates from each run. So here, 3 replicates showing you the N-linked glycans glycans at N149. And you can see across 3 replicates, they're very similar in abundance levels. And here's an example of one of the O-links at position 8 in the protein. So next we want to compare what is the actual advantage of doing HCD and ETHCD. And so here's the data from N-link glycan workflow. The first Venn diagram is showing comparing the total, the glycan compositions identified from both methods. So with ETHCD, we only actually identified an additional 14 glycan compositions, and an additional 40 glycopeptides were found. And at the O-link level, again, the vast majority of the glycopeptides identified were identified by the step collision energy HCD method. However, ETHCD data did add an additional 132 glycopeptides, which is fairly significant. And so the point of showing this is not the fact that we need to do ETHCD to drastically increase the identification numbers, but actually ETHCD is needed in order to get more accurate localization of these glycans, especially for O-link glycans. So on the left is the protein coverage view resulting from that N-link workflow and using the HCD method. So here we're only able to confidently localize one of the O-link glycans in this region of the N-rel protein, whereas using the IMPA and TRIP digest in ETHCD mode, we can confidently localize 3 additional sites within this region. And this also raises the question, maybe we should use IMPA cleavage specificity as part of our scoring algorithm in localizing these glycans, since maybe a little hard to see here, but a lot of these actually, the IMP enzyme was able to cleave directly upstream of this serine residue, as well as this serine at 199. And here's the evidence for that localization data. So here's glycosylation site brenine at 146. And I highlighted this B6 ion here, still associated with the glycan moiety, which we're able to differentiate these glycosites. So the B6 would allow us to localize the glycan to this threonine at position 6 in the peptide and exclude the other serine and threonines within that peptide sequence. And then likely here's a serine glycosite where we have a C4 and a C5 glycan fragment ion supporting that site and not the other ones within the peptide. And then lastly, another one where we have Y9 and Z6 glycan fragment ions supporting the localization of this serine rather than any of the first 3 residues in the peptide. Okay, in another study we did with purified glycoproteins within the BSI lab involved the CD33 protein, and we simply asked the question whether different host cell expression systems result in significant differences in glycan profiles. And so this lends to the fact that when purchasing or when studying glycoproteins, we need to carefully consider whether, which host cell system we're expressing it in, because different glycoforms can affect the structure and function of the protein. And so we used our N-link glycan workflow, and as you can see from all these purple bars represent different N-link glycopeptides. So we had very good coverage across the 6 different N-link glycosites in CD33. This one was produced in CHO cells, and here the other one was produced in HEC293 cells. And then we did site profiling and compared the glycans identified between the 2 cell systems. And we found a significantly higher abundance of fucosylated glycans at this particular position in CD33 from expression of HEC293. And also using the site profiling tool, looking at each of the 6 N-link sites, we wanted to identify the most abundant glycoform from each sample. And then we wanted to see whether we can match it to the intact data in order to confirm this finding. And so we added up the masses of all the most abundant glycans identified in GlycanFinder from peptide digests, and came up with the theoretical mass of all 6 N-link glycans, added it to the mass from the amino acid sequence of CD33, and compared it to the measured mass at the intact level, and we were able to match to the the highest peak of the intact data, and with an error of just 1 Dalton. And so this was a nice validation showing nice consistency between the bottom-up data and identifying the most abundant glycans in GlycanFinder and matching to the most abundant glycoform at the intact level. And lastly, I'll show you some enrichment data that we've been working on in the lab. It's still a bit of a method we're developing, but we've made some significant progress on this front. And so as part of our enrichment workflow, we do reduction alkylation with TSEP and IAA, do multi-enzyme digestion, desalting. Then we perform peptide quantification to ensure we're using the same amount of peptides for enrichment across each sample. So we have these columns that are packed with a cellulose resin. We perform activation and then add the desalted peptides, 200 micrograms to each column, perform a number of wash steps with acetonitrile and TFA, and then elute with MS water. And so from human cell lysates, we did 2 replicates from each. And so you can see the first replicate, we've identified over 1,700 glycopeptides, and then over 1,100 in the second replicate, and very few are left in the flow through. And from yeast enrichment data, 226 glycopeptides, but actually just recently we were able to significantly improve upon this number. We just don't have the data ready to present yet, but as you can see, we've significantly enriched the sample for glycopeptides, and there's very little that you can identify in the flow through from this experiment. And here's some screenshots from the glycoprotein coverage view. So after enrichment from the human cell lysate, you can see one of the more highly abundant envelope glycoproteins has very good coverage of these N-link sites. And then the column flow through has virtually no glycopeptides that map to the same sequence. And then from the yeast data, again, one of the more abundant glycoproteins, we can identify a number of N and O-link sites. As you can see here, we ran 3 different types of data all together. The orange bars represent glycopeptides from the enrichment data. Green and purple or pink would represent from the flow through or the input. So there's for this protein, there's no glycopeptides that map from flow through input data to the same protein, indicating that the enrichment is working well for these samples. Okay, I just want to thank everyone for joining the webinar today and listening, and I want to thank our team at BSI. It's really been a collaborative effort between our software developers, our application scientists, as well as our sales team. And at this point, I'm happy to take any questions that you might have.